Here is the flocculus here, and we'll make a cut towards this region here. Here we cut through that middle cerebellar peduncle, and here the inferior cerebellar peduncle like this. Then we'll turn it around again and cut the superior cerebellar peduncle up here. That is the superior cerebellar peduncle. There we go. And then we can lift up the cerebellum like this. Cut a little bit further there and there, here. And what we'll do first, take a look here at the cut surface here. Take away a little bit of the blood vessels. And now what you can see, here is the coral plexus in the fourth ventricle, here in the midline, like that. We'll take that away. And then you can see here, here is the flocculus, like that. We'll lift it up so you can see it better. Here it is. That's the flocculus here. And in the midline, here is the nodulus. And there is a connection here between the flocculus laterally and the nodulus in the midline. Flocculonodular lobe, which is primarily related to the vestibular system. Now, let's go to the specimen here and look at the rhomboid fossa here, and take away a little of this blood vessels, and we will make a nice cut here through the superior cerebellar peduncle. And as you can see, here it is, the floor of the fourth ventricle. Beautiful stria medullaris here crossing straight across. But to be able to see a little more details in the rhomboid fossa, let's go to a photograph of this specimen. This is the brainstem with the cerebellum removed. Look here on the cut surfaces of the cerebellar peduncle. Here's the middle cerebellar peduncle, and in yellow, the inferior cerebellar peduncle and the superior cerebellar peduncle. The floor of the fourth ventricle is shaped like a rhombus and referred to as the rhomboid fossa. There are two longitudinally running ridges, the median eminence, one on each side, of the median sulcus in the midline. Lateral to the median eminence is a somewhat ill-defined sulcus limitans. Some prominent fiber bundles can usually be seen running transversely in the floor of the fourth ventricle, and they are referred to as the stria medullaris. The stria medullaris here were for a long time thought to carry auditory impulses, but they do not belong to the auditory pathways. They connect the arcate nuclei on the surface of the pyramids with the cerebellum. Here is facial colliculus in orange. That's not the area of the nucleus of the facial nerve, as you know. It's formed by the underlying abducens nucleus and the genu of the facial nerve. In front of the facial colliculus is a bluish-gray area, the lucus ceruleus, or the blue place, which is usually easy to identify. The cells in the lucus ceruleus are noradrenergic, and they project to wide areas of the brain and spinal cord. Down here in the lower part, here is the hypoglossal trigone in red, and it's formed by the nucleus of the hypoglossal nerve. And the vagal trigone in dark color here is formed by the dorsal nucleus of the vagus, the largest of the parasympathetic nuclei in the brainstem. And here, laterally, this area is referred to as the vestibular area, and it indicates the location of the vestibular nuclei. Now we'll take another look at the intracerebellar nuclei, in which most of the cerebellofugal projections arise. We'll use this half brainstem cerebellum preparation, and we'll cut a nearly horizontal section along this line from the horizontal fissure that you see here towards the fastidium, or the top of the fourth ventricle. And you know, here you need not only imagination, but also a little bit of luck. Here we cut it. So, here is the cut surface of cerebellum, mesencephalon here in the lower part of the figure. Well, what a display. Look, the bag-shaped dentate nucleus, beautiful. You can even see some of the fibers coming out here, forming the superior cerebellar peduncle. Here's the fastidial nucleus, close to the midline, and just very close also to the fourth ventricle. And if you have sharp eyes, here is the emboliform nucleus and maybe some of the globose nuclei here. This concludes the gross anatomical dissection of the brain, and you will now be able to talk more comfortably about the structure of the human brain.